I think our next talk is a virtual talk from Kristen Fernandes, who's a PhD candidate at Curtin University, who's going to talk about DNA metabarcoding and its application for restoration monitoring.
All right, so hi everyone, my name is Kristen Fernandes. Today I'm going to be talking a little bit about DNA metabarcoding and how we can use it as a tool for biomonitoring and ecosystem restoration projects. So when we talk about monitoring, I think the national standards say it very well. We need to monitor because it's a really important part of establishing whether or not we're actually conducting successful monitoring, whether or not we're achieving the goals and proving to stakeholders that we're achieving the goals, and also to answer specific questions that we have about what particular treatments we're applying, whether or not it's returning processes or organisms to ecosystems. So monitoring is quite important. And I think Dave talked about it a little bit earlier, but fauna monitoring in restoration is quite poor. We see it more inclined towards vegetation monitoring. And if we're monitoring fauna, it's very project specific. So different particular species are targeted. So I think malleefowl would be a good example, but also in different projects, they only look for certain animals or certain taxonomic groups. And especially when we're looking at whole ecosystem monitoring and monitoring for function across entire ecosystems, the effort undertaken is very species specific. So you don't see a lot on everything in the ecosystem. It's across a very specific taxonomic group the effort is put into. So when we think about fauna and the amount of ecosystem functioning that is linked to fauna diversity, we see that they're responsible for a lot of stuff that, plants thrive on. So soil modification, seed dispersal, pollination, prey regulation, disease spread, I could go on. But we routinely overlook them in restoration monitoring, and especially for invertebrate groups. And I think part of what prevents us from actually going through and monitoring closely is that the amount of effort that we have to put in for doing this whole ecosystem monitoring. It's very hard, especially, if we're looking at vertebrates, we're going across multiple seasons, different trapping regimes, harsh terrain, and often with cryptic or trap-shy animals. So you're struggling there. And then when we're looking at invertebrates, which are very underrepresented in monitoring, it's an issue of effort in terms of it's easy to get animals in the trap. But then when you have hundreds of specimens, having to identify every single specimen is quite daunting. You need a lot of taxonomic expertise and a lot of time, which is why we're moving towards looking at genetic tools to help identify animals and help us monitor. And one of those tools is DNA metabarcoding. And DNA metabarcoding is a very useful tool for rapid biodiversity assessment. So a little bit about how it works. We have our samples that we collect, and some of these samples can be arthropods, so you can look at full insect traps. You can collect soil, water, scat. There are some studies that are even looking at air, so taking filters from air filters and looking at the DNA that they have on them. You extract the DNA out of the traps, and what you do is you then amplify it using PCR amplification. So that's a process of heating and cooling, and what it does is it creates multiple copies of DNA. The beauty of metabarcoding is that you can put through hundreds of samples at a time, because this PCR amplification also includes the identification tags that are added on to every sample. So when you get to sequencing, instead of looking at one particular sample, you can sequence hundreds of samples, and all the sequences, the DNA sequences, are individually tagged and can be referred back to the original sample that you looked at. So all the taxa can be looked at. And when you're identifying those sequences, you look at a reference database. So these are publicly available databases where people have stored barcode data that matches back to the species you're looking at, and you get an identification for what was in your sample. So very useful tool. And essentially what you get is a tube of clear liquid containing DNA that also contains a breadth of biodiversity. So not only do you get the animals so from an insect trap, not only would you get the animals that you're looking at, but you'd also get the plants and the other animals that they're interacting with. And essentially where we want to take this is into looking at whole ecosystem biodiversity. So from the microbes to the vertebrates, invertebrates, plants, we can look at predator-prey interactions, disease interactions, everything be looked at simultaneously, and we get a much clearer idea of how our restoration is progressing. So it is a very useful tool. And we've had a few papers come out recently, so some of my papers, but then also across the world that have used it for restoration monitoring. So there have been some in the Amazon and the tropical forests in China, but also in Tasmania and things. So this technique is being used for restoration monitoring quite a bit. People are testing it out and seeing how it goes. And of course, let's talk about some of these advantages, because it's completely non-invasive. You don't have to go out there, you don't have to tag animals, you don't have to trap animals, you don't have to cause them any distress. And with multiplexing hundreds of samples at once, you can gain quite rapid results. And because you're only looking at the story that the DNA is telling, it's highly standardizable. So across projects, across a single project, when you take a sample, you will be processing it in the same way. The same set of conditions are applied to it. You're not looking at it through individual lenses. It's all through the same pipeline. So highly standardizable, which is very good if we're looking at meeting regulations and stakeholders' expectations. And because you're only letting the DNA tell the story of what's there, you can detect these cryptic or difficult to detect species because they leave their traces around, right? Some of the challenges though is that you can get some false positives occurring. So whether that be from the PCR process, the sequencing process, you get some errors, but then you can also get some contaminations. So species that are not really meant to be in the site will show up, and it's hard to tell sometimes whether or not this is a real signal or if this is a false positive. At the moment, we can't really accurately figure out species abundance from metabarcoding data alone. There are some alternatives, so you can look at relative species abundance. You can use just the number of times they occur in a site and then work out how many species there potentially are. There are some new methods, so spike pipe, which came out of Doug Yu's lab in China, where they have an internal calibrator, so you're working off biomass, so you can work out how much of one species in a sample. But this is still in the development stage. We still haven't gotten to the point where we can use metabarcoding to accurately determine species abundance. Additionally, with false positives, you also get some false negatives. So if your species isn't in the database, even though it's at the site, if you're using a database that has not been updated, has not got that species in it, you're not going to detect it. And that's a real problem, especially when your study is looking for full biodiversity. You want to have an accurate representation of what's in the site. Additionally, if you've chosen a primer for PCR amplification that's not matching that taxonomic group, not matching that species is not going to show up. Even though the DNA is in the sample, this animal has been observed at the site, it's not going to be there, which can be quite problematic. So in the sense, DNA detection doesn't always mean that the organism is present, but non-detection of the DNA doesn't always mean that the organism isn't present. So there are still some unknowns. So we don't really know how organisms shed DNA into the environment. We know that mammals will defecate, will urinate, they'll shed heaps of hair everywhere. So sometimes they can be easier to pick up. But when you've got reptiles that don't defecate as quickly as much, they don't shed as much DNA, they can be harder to pick up in our samples. Also, we don't really know what conditions impact DNA detection. So we're aware that high UV, high temperatures can degrade DNA. So if you're in an arid environment, you're collecting samples from the surface of the soil, the UV has interacted with it, there's not going to be a lot of DNA present. But if you're going into Arctic conditions, very cold, stable environments, the DNA could be preserved for years. And there have been studies that have pulled out mammoth DNA. So it's hard to tell in those cases if the animal was present in that last week, or if that animal was present maybe a year or two ago and the DNA is just really well preserved. So there's some unknowns and some issues. And today I really wanted to talk about this case study that we used to study invertebrate assemblages in restoration sites and whether or not they were returning back to what we expect in a reference community. And this was a kind of our first foray into using DNA metabarcoding for restoration monitoring, and something that we came across some of these challenges and how we overcame the same sort of issues that I've been talking about. So we looked at 2 sand mines in Western Australia. These sand mines, I think they were restored through topsoil reapplication. So they had stored the topsoil, and once they had finished with the pit, they would reapply the topsoil. I don't know if they had specific plantings as well, but they really weren't monitoring much fauna. I think some birds, because this is Banksia woodland, they were trying to return it to. And so we applied metabarcoding to insect pitfall traps. So any ground dwelling organisms, they're ground active. So they would be walking around, they'd fall into our trap. And the first thing that we realized was that the assays that we had chosen to use, that a lot of people had used, were not picking up a taxonomic group that was present in 90% of the samples that all of us could see visually in the samples, and that was ants. And that was a huge problem. So we went back and we designed ant-specific assays using a barcode reference database created from Pilbara ant species, and we were able to create this specific assay for this project. We looked at core restoration ages. We looked at some really old restoration, which is about 13 years since the mining, and some younger stuff, so only about 2 years. And then we also had some reference vegetation sites. So essentially what they were trying to take it to was this pristine Banksia woodland habitat and this like bare, sandy pit, essentially. And they were looking at vegetation. Vegetation was being monitored, so we were looking at established vegetation plots. So you can see in that third photo from the left, there is that pole there. So they had vegetation monitoring plots that they were monitoring. So we were just going off what they had used previously. And what we found was that there was a distinct trajectory and a significant trajectory of insect communities from this younger restoration to assembling more closely to the reference sites. But there was one clear indication that this restoration was probably never going to be exactly the same, was that there was this disturbance specialist ant species. So the green-headed ant, Reticularia metallica, showed up in every single one of our restoration and vegetation sites. So even after 13 years, there were signs that the site was still, that site was damaged in the past. So whatever we do, it still leaves scars on the landscape. We also found in these younger sites, there were lots of springtails. And if you know anything about springtails, you know that they're really great soil architects. And they work really well to cycle nutrients, aerate soil. So it's a good sign that they're occurring in these younger sites, because you can see that there would be impacting the function and increasing the chance of function returning to these systems. Something odd we noticed in our reference sites was that there were a lot of these Julida millipedes, or the Portuguese millipede, and they were showing up everywhere. And if you've lived in Perth, you know that these Portuguese millipedes are a pest and they can cause train derailments. They assemble in such masses, but they are detritivores, and they love humus-rich soil. They love lots of vegetation to grow in, and so they were present all in our reference sites and in our 13-year-old sites. So the sites were creating a habitat for these millipedes to thrive in, even though they were not probably what we would have wanted to thrive in our reference bushland. We need to think about when we're doing metabarcoding is that even though we can get these really cool results, these results can tell us a lot about our environment, it's not a silver bullet. It's just another tool that we can put into our monitoring toolbox, and we should use it as such, right? So what do we need to think about if you're wanting to undertake some metabarcoding work? So the first main question you need to ask is, what are the outcomes required for your monitoring program? What species do you want to detect? How in-depth do you want this to be? Do you want to know everything in there? Do you want to know only certain groups? Because genetic studies can be expensive. Although you're saving time and effort, these genetic reagents, they do run up the cost a little bit. But I mean, we live in a capitalist world, things are getting cheaper every single day if there's more demand for it, right? So even though the funding may decrease, there is still, in the future, there is still that question of, do you have enough funding to undertake such studies? And to design a good study design, essentially. You wouldn't take soil samples if you're looking at you're looking at flying insects. You'd want to collect the right sample, the right design, the right study for it. It's very important to think about the questions that you're asking and see how metabarcoding can fit into answering those for you. So in conclusion, biomonitoring will always rely on a combination of approaches. There's rarely, I think maybe in the future, we'll be able to walk out into a field and have a radar gun that'll tell us exactly everything that's there. But at the moment, there's not one size fits all. It's always going to be a combination of things. And genetic tools like metabarcoding will be more prevalent in the future because it increases the accessibility of our ability to monitor fauna. So it's a very cool technique, a very cool tool that we can use, and I know that it's going to be used a lot more in the future. So I've got some further reading, some further sources if anybody wants to check these out. So these are some studies from my lab, so the Trace and Environmental DNA lab, but also some work being done out of the Centre for Mine Site Restoration. And I've also put in my Twitter and my email if anyone has any more questions. I'd be happy to take them. Thank you.